Hello everyone. Happy Women's Day to all of you. Myself, Dr. Raman Singh, International GWIS member. Let me introduce myself. I have a teaching and research experience of more than 9 years, which includes National Dope Testing Laboratory, New Delhi, which is WADA accredited lab, some NAC A-grade universities. Currently, I am working as a senior research scientist, WTL Clean and Renewable Energy Private Limited, New Delhi, India. My research interest includes organic chemistry, including medicinal chemistry, synthesis of some biologically important heterocyclic compounds, drug resistance, traditional knowledge, IPR, intellectual property rights, and green chemistry. My academic identities are displayed in these slides. I have published total 18 research papers, in which 16 are Scopus indexed with the highest impact factor of 4.3. Total book chapter published are 5 and book published are 4. I am member of different professional bodies and member of advisory boards and editorial boards. My research focuses on synthesis of biologically important heterocyclic molecules. Today here in the international chapter meeting, I am discussing one of the important pharmacological active moiety, that is beta-lactam. So the beta-lactam core represents a 4-membered heterocyclic ring amide as displayed in this slide. The beta carbon and nitrogen linkage form the heterocyclic ring, and therefore the heterocyclic system is named as beta-lactam. The simplest beta-lactam is known as 2-azetudenone. There is a need to design and develop new molecules which are either act as an antibacterial against AMR bacteria or deactivate the mechanism of antimicrobial resistance, enabling the old drugs. So this beta-lactam core is very important in pharmacological activity and established a very good structure-activity relationship. This beta-lactam core or moiety is present when it is present in any this molecule. It enhances the pharmacological activity. This beta-lactam core is associated with different pharmacological activity like anti-tubercular activity, antimicrobial activity, anti-cancerous activity, anti-HIV activity, or enzyme inhibitory activity. So literature survey reveals that the beta-lactam is a very important pharmacological core. So looking to the importance of this beta-lactam moiety, our research strategy includes the synthesis and characterization of precursor, synthesis of key compounds by incorporation of N-heterocyclic biolabile ring at azomethane linkage, and antimicrobial screening of newly synthesized molecules. So synthetic strategy involves the preparation of precursor having this azomethane linkage, and further this beta-lactam moiety, which is a very important pharmacophore, this is incorporated at this azomethane linkage to form a pharmacological active compound. So how these precursors are formed, or which strategy includes the generation of azomethane linkage. So substituted aldehyde when reacts with amines gives the different substituted compounds which is having this azomethane linkage. So the product formed or the precursors formed are oximes like hydrazones, imines or semicarbozones or thiosemicarbons. Now these imines, oximes and hydrozones, they are ready with the linkage, azomethane linkage, and the beta-lactam core is incorporated at this azomethane linkage to form or to enhance the structure activity relationship of the particular compound. So in our research we are trying to synthesize some new active pharmacological compound. There is a big problem of drug resistance. So antimicrobial resistance, it is emerged as a major challenge for current medicine and healthcare system. It is a serious problem in healthcare settings or systems, overuse of antibiotics. It developed antibacterial resistance, and WHO recognized this as one of the major problem. Without effective antimicrobials, the success of major surgery and cancer chemotherapy would be compromised. So drug resistance is a major issue. See, if a compound is a biologically active drug, so if there is a resistance for the species, it becomes inactive. See, if we use some activators, it can again be pharmacological active, but the drug resistance is still a major issue in the healthcare system. So what we are doing, we are trying to synthesize some new pharmacological active compound so that we are ready for the new candidate drugs, new active pharmacophores containing beta-lactam derivatives which can have a good pharmacological activity. So the expected outcome of our research work is that we wanted to establish a synthetic methodology to generate a new class of hybrid molecule from the known pharmacophore responsible for antimicrobial activity, and the antimicrobial screening of these new chemical entities may result into some new significant molecules. So we are trying to synthesize some novel beta-lactam derivatives. As I told you that beta-lactam is a very important pharmacophore. So we are trying to synthesize some novel beta-lactam derivative which will definitely have a great impact on pharmacological and chemical knowledge and find value in health and medicine. So in view of exploring this microbial infection, the significance of a research work in Indian context and for all human welfare is high. So we synthesize organic molecule, we try to synthesize some biologically active compound and open for the collaborations for further antimicrobial studies in vitro, in vivo, like toxicity. We are ready for the collaboration in bioinformatics, QSAR. We are ready for the collaboration in our autodoc or chemoinformatics studies or any other biological activities. I am ready for the collaboration, and I am providing you the molecule or the novel compounds, or you can do the in vitro, in vivo, any biological activity for that. So finally, thank you. Thank you organizers for providing me this opportunity, and thank you once again. Happy Women's Day once again. Happy Women's Day to all of you. Thank you.